In this video, we will be discussing another very important part of photosynthesis, which is known as chemiosmotic theory or chemiosmotic hypothesis. And this is going to help us understand how exactly ATP is synthesized in cyclic and non-cyclic pathways. We have seen that when the electron passes through the electron transport chain, it releases that energy and that energy is used up in synthesis of ATP. In that pathway, we just write it in a very simple manner, though the actual process is very much different from what we represent there. So, chemiosmotic hypothesis is actually helping us understand that. So, let us draw one thylakoid and see what all is present where, so that we can understand the exact process of this ATP synthesis. This is the thylakoid membrane, and we have drawn two membranes. That means there is an outer membrane and an inner membrane, and this space which is inside is the lumen. So this is the outer thylakoid membrane and this is the inner thylakoid membrane, The cavity inside is known as the lumen. Two things which we have already discussed. On the inner membrane of the thylakoid is present a complex which helps in photolysis of water. So, and we know it is present on the inner membrane. So, this is the complex for splitting of water. That is photolysis. And the next thing that we know is the presence of an enzyme. So, the enzyme which is known as NADP reductase. So, this is our NADP reductase, which is present on the outer side. If this is the thylakoid, then the things which are around it, thylakoids are present in stroma. So, what is present around it is stroma. So now we know exactly what is this structure. There is a membrane. The membrane has an outer surface and an inner surface. There is a cavity inside which is called the lumen, and outside thylakoid is present stroma, the matrix in which these thylakoids or grana are placed. The complex which is going to help in splitting of water is present on the inner surface of the membrane, and enzyme NADP reductase is present on the outer side, that is towards the stroma side. Let us draw the photosystems. The photosystem, that is PS2, is located here and PS1 is located here. And we have discussed this, that is non-cyclic photophosphorylation that we are talking of. The electron here is going to be the reaction center chlorophyll. At the top will be the primary electron acceptor. And then the electron is going to pass from PS2 to PS1. And here is going to be the electron transport chain. We wrote 3 electron acceptors or the molecules through which the electrons was being passed on. There was plastoquinone, then was cytochrome, and then plastocyanin. Here we are drawing only cytochrome. So say this is the cytochrome which we have drawn here. Now what exactly happens in non-cyclic photophosphorylation? The same thing we are representing here in a slightly simpler and a different manner. When sunlight falls on this, when sunlight falls on PS2, all pigments are going to absorb the photons, will pass it on to the reaction center chlorophyll. the electron pair will get excited. It will be taken by the primary electron acceptor, and now this electron pair is passing through the electron transport chain. We said that this electron, when goes from higher level to lower level, it releases energy. So at cytochrome, it is going to release that energy. That means when this electron is passing through cytochrome, and finally it will go to PS1. But when this cytochrome is taking electron from plastoquinone and passing it to plastocyanin, at that time, cytochrome accepts a pair of proton from stroma. It has accepted a pair of protons from stroma, and using this energy, it pumps that pair of stroma into the lumen. That means the energy which is released by the pair of electrons, when it is coming down the gradient, that energy is used by cytochrome to accept a proton from stroma and pump it into the lumen. So actually, what is a cytochrome doing here? It is accepting proton and transferring electron. So it is actually a proton acceptor and an electron transporter. Though we call it an electron acceptor, in electron transport chain, we are saying that these are all electron acceptors. But this cytochrome is actually an electron transporter. So when it takes electron from one, passes it on to the next. During that passage, the energy which is released is used up to pump a pair of proton which it has accepted from the stroma into the lumen. This is part 1. Secondly, to replace that electron pair, photolysis of water is going to take place. So now this water splits here. The complex which is helping is on the inner side. So splitting of water will take place in the lumen. 3 things are released. A pair of electrons which is given to the PS2. A pair of protons and half O2 or 1 atom of oxygen, which is the byproduct. So this byproduct, that is oxygen, is produced in the lumen, and then it is going to diffuse out and will be given out as a byproduct. But the site where oxygen is synthesized or produced is the lumen of the thylakoid. Oxygen will diffuse out and will be released. Electrons are given to the PS2, the chlorophyll, that is reaction center chlorophyll of PS2, because from here the electrons are continuously being emitted because of that high energy. Now the only thing which remains here is the proton. So now the protons which are produced due to photolysis are also in the lumen, and the protons which are being pumped from the stroma into the lumen, they are also accumulating here. This results in accumulation of protons in the lumen and reduction in the level of proton concentration in the stroma. So there are 2 sources from where the protons are coming into the lumen. One, from photolysis of water, and second, by this electrons moving through cytochrome. The energy which that electron releases, cytochrome accepts protons and pumps it into the lumen. So lumen has high concentration of proton. So there is a gradient again, high concentration in the lumen and low concentration in the stroma. These protons would tend to come back into the stroma from higher concentration to lower, and they can come out only through certain particles. So here there are some proteins. These proteins are known as F0 particles. These are integral proteins, embedded proteins. That means they are embedded in the plasma membrane. And there is again a protein which is extrinsic protein, which is protruding out of the membrane, or you can say it is protruding into the This is known as the F1. And this F1, it acts as ATPase. It acts as an enzyme for the synthesis of ATP. The protons, they can come back into the stroma only through these channels. So these F₀ and F₁ particles, they are actually acting as channels for the protons to move from lumen to the stroma. And as the protons are moving from higher concentration to lower or through a gradient, they would also release that energy. So now when this proton pair passes through these particles, the F₀, it helps in facilitated diffusion of these protons. F₀ particle, which is an integral embedded protein, it helps in facilitated diffusion of protons. That means it is going to bind with the proton here, it will move and come here. But as we are using the word diffusion, there is no energy required. And now when these protons, let me make a line which is wavy, that indicates that they are coming with energy. And that energy is used by this particle to add inorganic phosphate to ADP and synthesize ATP. And after this energy is released by the proton, with that minimum energy, it comes back to the stroma. So now what exactly is being done in the light dependent step, that is cyclic and non-cyclic photophosphorylation? Because we are talking of this thylakoid, we have taken non-cyclic photophosphorylation. What has happened in non-cyclic? Light falls on PS2, electron pair excites, it passes through electron transport chain, goes to PS1, again it will be taken by NADP. We will continue that and we will finish that in a minute. But in this first part, what has happened is that when the electron pair was passing from PS2 to PS1 through cytochrome, cytochrome accepted a pair of proton and pumped it into the lumen. Photolysis also is taking place of water. That also results in generation of protons here. So there is high proton concentration in the lumen. These protons can come out only through channels. These channels have two parts. F₀, which is going to help in facilitated diffusion of protons, and F₁, which is acting as an enzyme for ATP synthesis. So when proton is coming down the gradient, down the concentration gradient, it releases its energy. That energy is used up by this enzyme or F₁ particle to add the inorganic phosphate to adenosine diphosphate. It becomes a triphosphate, that is ATP is synthesized. If you are able to recall the way we made non-cyclic photophosphorylation, we started with PS2, then we said this is the primary electron acceptor, and we made these three electron acceptors. This was cytochrome. And then the electron was sent to PS1 and again here, and then it was given to NADP. We just made ATP here. We made ATP. We just said that ADP and inorganic phosphate, they combine and we are able to synthesize ATP. So in this process, it is difficult to place this thing there. But ATP is not synthesized like a simple process. For ATP synthesis, there has to be a proton gradient which needs to be generated. And when these protons come down the gradient, that is the time when ATP is synthesized. ATP is synthesized in chloroplast also and in mitochondria also. In mitochondria also, ATP synthesis is associated with proton gradient, and here also it is associated with proton gradient. Now let us complete this process. The electron pair which is coming here will go to the primary electron acceptor of PS1, and then through electron transport chain, this pair of electron will go to NADP, and this enzyme NADP reductase is going to help in formation of the reducing power. But for that, proton is required. From where are we going to get the proton, or from where is this proton made available? These protons are coming back here into stroma. So the protons which are coming here, and there are many such protons which have come back. But to understand this, say this is the proton which is joining here, and these protons and NADP in presence of this enzyme are going to give us NADPH2. That is our reducing power. So this entire process is known as chemiosmotic hypothesis or chemiosmotic theory, which explains that during photophosphorylation, when ATP is actually synthesized, the process is not exactly we just tried ATP synthesis here. At cytochrome, protons are being transferred, and when these protons come back into stroma, that is the time when ATP synthesis takes place. This is very, very important. The points which we have to remember in chemiosmotic theory. Number one, the photolysis complex, or the complex which helps in splitting of water, is on the inner side of the membrane of thylakoid. NADP reductase enzyme is on the outer side. This is going to help us understand where is what produced. Oxygen is released in the lumen, and NADPH2 is produced in the stroma. ATP is synthesized when the protons come out. So ATP is also produced in the stroma. And if you are able to recall the first introductory part of photosynthesis, we said light reaction takes place in thylakoids or membranes, and dark reaction takes place in stroma. And for that dark reaction to take place, three things are required: carbon dioxide, NADH2, which is the reducing power, and ATP. So all these three things will be made available in the stroma itself, and stroma has all the enzymes to help in dark reaction. So the points which we have to remember: site of water splitting complex, that is inner side of the membrane, and the membrane is of thylakoid. The next important thing is site of NADP reductase enzyme. It is the outer membrane. Next important thing, site of oxygen production, that is lumen. Next, site of ATP and NADPH2 production, that is stroma. This is stroma. And one more thing which we have to remember is about the cytochrome. This cytochrome is not exactly an electron acceptor. It is a proton acceptor and electron transporter. It is transporting electron, and during that transportation of electron, the energy which is released is used to accept proton, and that proton is now passed on from stroma to lumen. So this process is the exact process by which the ATP is synthesized during photophosphorylation.